Ed Buckler from the States. There we are. I'll find your presentation for you.
Yeah. If you were good at Word every day.
Yeah, I've avoided the error that everybody else was making this morning. We blame Nick for this morning, making everything go wrong, but Brian wasn't much use at putting it right, so there we go. Anyway, here's Ed.
Thank you very much. So first off, I want to thank the organizers and thank Brian very much for inviting me to the symposium. And it's been a lot of fun already to chat with folks. So I want to give you a little bit of a perspective of where we're coming from the plant breeding and plant genetics side of things. Given that, of course, Fisher has essentially dominated our field for the last 100 years very successfully. The work I'll be telling you about has been supported by the USDA and from the U.S. National Science Foundation. And then I'll be talking about some very large empirical projects. And these have been the efforts. I've now collaborated with nearly 1,200 different people across the globe in order to do some of these experiments. And so really what I'll be telling you, a lot of the empirical results have been by collaborating and learning from these wonderful people all across the planet. So I have a background in also not only genetics, but also in archaeology. And so whenever I start thinking about crops and domestication, I always want to start 10,000 years ago to where were the various stages of plant breeding. Well, 10,000 years ago, we were essentially, I would say, applying breeding 1.0. We were applying phenotypic selection. We were figuring out which of the 250,000 plant species out there could be domesticated and which ones didn't kill us. And we were then moving those on and selecting some of them very intensively. Then a little over 100 years ago, really controlled trials come into being, quantitative genetics, experimental design, statistics, and equipment like harvesters that give us very accurate measurements of phenotype come into being and really start driving plant breeding. And then a little over about 20 years ago, and Mike Goddard helped bring in the genomic selection revolution, where we were able to start doing whole genome prediction and making progress there. But of course, those breeding 2.0 and 3.0 really do spring directly from Fisher. And whether it's how we measured our plots, how we organized some of the statistics, and analyzed the data out there to now genomic selection. Have all come from that infinitesimal model and are driving things. And so I'll do a little bit of a flashback in my own career and how we kind of looked at this. You know, I started working on maize, and part of why I found it very interesting is it was so high diversity. Any two maize varieties are as different from one another as humans are from chimpanzees. So in many regards, you can start asking the question, what does the quantitative genetic variation look like in a species that's this diverse? And so, you know, at the time, and now we actually can see this, there's functional variation at every gene. And so what does the genetic architecture look like when you have hundreds of thousands of functional SNPs across the genome? So the hammer we used to first look at that was we took 25 lines of maize that were as diverse as we could find on the planet. We crossed them to a reference line, and we created 5,000 lines derived from those. And then between ourselves and our colleagues across the globe, we planted them out. We measured them for lots of different traits and so on, and mapped. And many times we've actually mapped various genes controlling various traits. But what I want to highlight for you is that how infinitesimal was it? Well, if we just look at how those various regions of the genome and the various donors donated to the resulting lines we were able to produce, 94% of the genome was within 4% of 50-50. But actually, we would have expected even a tighter distribution if there had been no differences in fitness between each of those. Turned out that actually 59% of the genome exhibited substantial departures from that expectation. So we weren't seeing massive fitness effects all across the genome, but we were seeing lots of subtle fitness effects just in the production of those lines when we made all those crosses. We also then looked at traits, for example, flowering time. This is probably the trait that has been under some of the strongest selection as maize moved from central Mexico to North and South America over an incredible latitudinal climb. And maize now flowers in anywhere from about 45 days to 125 days. And so when we did QTL mapping and association mapping for these types of traits, what we found out was even though they had that tremendous phenotypic range, the largest effect QTL we could detect was 42 hours, and 98% of them had an effect less than a 24-hour effect. It's small effects all across the genome. Many of these we've now cloned, we know what they are, and in near isogenic lines in a uniform background, they do have these effects. This was an accurate estimate of it. Flowering time is essentially the product of stacking up lots of little QTL all across the genome, early flowering going one direction, late going the other way. So, but we also could add up all those effects across the genome. And so this is the product of adding up all those little effects from training on 5,000 lines to predict the parents. And as you see, we can predict those parents very well. Now, this is not the way you'd want to do genomic selection. Training sampling 5,000 lines to predict 25 is a pretty stupid way to do it. And yes, we are sure there is epistasis out there, but essentially the simple models we did to add things up, it showed relatively modest departures. So Fisher was right. What do we do next? So, and I want to highlight that I think for most of plant breeding and where most species are across the globe, we should still be focused on breeding 2.0 and breeding 3.0. There's lots of species where this isn't true. But in the case of maize, people have been spending a couple $1 billion per year on R&D, and maize has kind of moved to a place, especially elite maize, that they've done a lot of really excellent breeding, and the question is, where do we go next? It's getting increasingly expensive to keep on doing breeding 3.0 to move things along. And this is where I think a lot of the excitement is, how do we use the editing technology that's out there, starting to identify the causative nucleotides and start really designing these genomes. When we talk about editing here, I want to highlight, we're not talking about making one or two changes. We're thinking about making hundreds or thousands of changes across the genome. So every generation in maize, we have anywhere about 90 mutations per generation. A few of them are adaptive, some are deleterious, and most are neutral. And if you add those up across a breeding program, we take one of these diverse maize line or diverse breeding populations of maize, there may be 80 million SNPs that are segregating out there among them. And we want to get down to that list of the few tens of thousands of ones that we think are functional, and then those are the ones we want to start tweaking. And we know that GWAS and genome-wide prediction type approaches aren't going to be the only way we can get down to the bottom of that funnel. And of course the problem is, let's take that NAM population, we create 5,000 genotypes, but we grew 1 million or a couple million clones out in the field. And so we have really good estimates of phenotype, but still only on 5,000 genotypes. And so we've got a massive NP problem of far fewer observations than we have parameters. And of course, this is very difficult to solve. So a lot of our community, what we're trying to now figure out is how to use these various filters to get from that top to the bottom as efficiently and cheaply as we possibly can. And I'll talk about a couple of those today, mostly about conservation and then how we're using machine learning to tap into some of those other areas. And the basic thing we believe here is that by using molecular traits in evolution, we don't have an NP problem. We actually do have more observations than we have parameters in these types of situations. And so for chromatin structure, we can actually learn how the entire genome folds up by using 2.3 billion observations that are coming out of our data sets, or expression. We have sampled expression of 70 million different alleles and genes across the genome, and we can start modeling how expression works for every gene in the genome without doing those. And so we're in a situation where we're not in that over-parameterized situation. And just to kind of give you a feel for what we were able to do and with those field trials from lots of different people's efforts, if with those 5,000 lines and lots of replication, we were able to map roughly about a 5-kernel difference per plant down to a gene. We could certainly attribute variance to a larger, much more finely than that, but the mapping resolution that got us down to a gene was about 5 kernels per plant. And just to compare what we can do with evolution relative to that, so evolution, of course, is really this integration of billions of years of field testing, And in the case of maize, just within its allelic variation, it's capturing about 3 million years of evolutionary time among those alleles. Jeff Rosser-Barr estimates an average of effective population size over the last few 100,000 years of maybe about 100,000. And so selection should be able to see a difference there of about 1 kernel in 250 plants, about 1,200-fold better than what I could do by growing a whole bunch of plants out in the field. Now, of course, the problem with using evolution is we don't know the conditions under which selection operated. We've already heard about it's pretty hard to figure out what connects those two together. But our ability to look for signals of selection have gotten much better with our ability to just sequence. And so we have a pretty good idea that maybe about 1,000 loci across the genome have been involved in domestication and improvement in the last 10,000 years. And so that's about 4% of the genome. The other 96% of the genome, we believe that pretty much the physiology of a plant hasn't changed in relative to its wild relatives versus the modern one. Photosynthesis fundamentally still works the same way today as it did 10,000 years ago. And so we think that using evolution as our guide helps us, for at least a lot of the genome, or deep evolution helps us out for a lot of the genome, not all of the genome. So let's see how we've been able to use evolutionary conservation to figure out where the deleterious mutations were. So deleterious mutations and inbreeding depression, of course, discovered long ago, and maize breeders were pretty clear that this was key to inbreeding depression about almost 100 years ago. Our challenge, though, today is to really find those deleterious SNPs and catalog them at scale. And so the question has been, can we catalog them well? So a postdoc in the group, Eli Rogers-Melnick, a few years ago, he lined up maize versus 12 other genomes that were available at the time and asked the question, base pair by base pair, how conserved is every base? And then using these group estimates, characterize each nucleotide. He found that only about 10% of the genome was alignable and constrained. And I want to highlight that this style of analysis at this time point, we were mostly able to work with protein changes. We weren't able to deal with regulatory changes because those regions of the genome didn't align. First reality check he wanted to see is, of course, in low recombination regions of the genome, you should have a buildup of deleterious mutations, and in high recombinations, you should have less. And that's exactly what he saw, is we had a buildup in the low recombination regions and fewer deleterious mutations in the high recombination. But I do want to highlight, we have deleterious mutations everywhere. It also looks like biased gene conversion is maintaining deleterious mutations, for example, in some of the high recombination regions at a fairly high rate. We then looked at a chromosome that's kind of special in maize. This is, this chromosome 5 in maize, for especially temperate germplasm, has always showed very high levels of heterosis. And so what we've plotted out here is the recombination rate in green versus the enrichment for deleterious mutations in black. And what you see is in the low recombination regions in the center of the chromosome, we get a buildup of these deleterious mutations. And that region in the paracentromeric region is also exactly where we see the greatest level of heterosis in maize across this chromosome. So where heterosis was lining up, where the buildup of deleterious mutations were lining up, appears to agree quite well. So we are able to use conservation to identify some of those deleterious mutations. But it hasn't been really, this type of approach wasn't helping us very well on the regulatory side. And we estimated that at least half the genetic variance is coming from regulation of the genes, not just the amino acid changes in the genes. And so this is where we've been starting to really shift and using machine learning tools to train off of those various molecular data sets. And today I want to talk a little bit about how we've been doing machine learning on chromatin structure and expression levels in maize. And so why are we using machine learning? Well, it processes massive amounts of data. There are very clever algorithms and hardware now that allow you to just process more data than you've ever really been able to in the past. It isn't that machine learning some, I must say, a couple years ago I thought this was mostly a gimmick, and it wouldn't be particularly helpful, but there are aspects of how it's been implemented. It's just extremely efficient from the computer science point of view. It also allows us to really generalize rules from a portion of the genome to the rest, and it is allowing us to take a lot of knowledge from a single genotype and apply it to all the rest of the genotypes. And because the models can be used then to also determine, we can tweak the model, throw in a new sequence, and determine whether or not that new sequence is going to disturb the model. So we can base pair by base pair ask for other effects we can measure. So the basic models we've created, we've started with sequence, and we ask, can we, using those sequences, predict is the chromatin open, what transcription factor binds there, is it a pseudogene, and what is the expression level? And then if we give it two homologous sequences, can we tell whether or not the expression of allele A or allele B is greater? And three great postdocs in the group have really been spearheading this: Catherine Mejia, Hai Wang, and Jacob Washburn. I want to highlight that we could absolutely get more accurate models that if we included additional annotations other than the sequence to make these predictions. But we really want to create a series of models that only use DNA sequence to make those downstream predictions. We don't want to have to rely on any other molecular biology annotations. And of course, the genome is figuring out how to do this just from DNA sequence. So we want to be able to get as close to that as we possibly can. We've been using two basic approaches here. One is a K-mer-based approach, and this is coming from natural language processing ways. So you type in something into Google, it's doing natural language processing on your words to figure out what you want out of there. And I'll show you how we're breaking it into a sequence. And the second approach is we're using convolutional neural nets. These are the class of models that are being used to differentiate between a picture of a dog and a cat. But we were applying those to DNA sequences to identify things. And so they allow us to process sequences at different scales, and then are allowing us to use these neural nets to integrate the process sequences. So to give you a little feel for how the K-mer-based approaches work, the simplest one is what we call a bag of K-mer model. And essentially, we just take the sequence, we break it up into its K-mers, we count them, and we fit them into a regression model. And so that's the simplest type of model. There are vector-based ones, which are keeping a count of them and the order of the words, so that you essentially are working more closer to like a grammar that's out there. And then the other class, the way we take the convolutional neural nets, is we take a sequence, we essentially are making a black and white picture out of it, and then we run convolutions over that. And what the convolutions are doing is they're summarizing those patterns at different scales, and then the neural nets pick up those summaries from the convolutions and then can relate them to a response such as expression levels. And so we've been using both those classes of models. And just to start with the K-mer-based ones, we looked at whether or not we could predict where the transcription factor Phi4 binds to in the genome. And we're seeing how well we can predict where these bind relative to other matched sequences that have similar types of GC contents and so on. And the model she creates, the shuffled models along the diagonal there, she's able to create a very highly accurate model with an area under the curve, or ROC curve of about 0.99. And it isn't just this one transcription factor. She can do the same thing for a wide range of different transcription factors, like NOD1, open chromatin measures, from like MNase. These are similar to what DNase or some histone marks would do. And then core promoters, she can predict very accurately using these K-mer-based models. Some of these models are transferable between one species and another. So for example, we've been able to train a model in maize for core promoters and asked, does it work in sorghum? And it sort of does. It doesn't work great. But her maize core model for NOD1, it translates over to rice almost perfectly. So there's certain classes that we can train in a very broad spectrum. Other ones, we certainly still need some work to get them to work across species and so on. There's been a lot of complaints about machine learning models that it can be black box, and absolutely that's true. We can act like these are total black box models. But for example, these K-mer models, we can open them up and ask what are the K-mers that are getting the heavy weights in them. And for example, this is a core promoter model, and so we would expect that, for example, the K-mers that represent the Tata box should be overrepresented there. And that's exactly what we see, is that K-mers that represent the Tata box are enriched right in front of the promoter, and so on. So I would say a lot of us are learning now how to open up machine learning models and make sure we're learning something mechanistic rather than just memorizing things that we don't want it to memorize. She's been able to extend this type of model to, she had data now from about 100 to 200 different transcription factors, and she created individual models for each one of those, and then she clustered the models and asked which models are most similar to one another, and the transcription factors that were essentially in the same family, they're clustering together. So it actually, where they were binding is replicated by what transcription factor family they're in. So we are starting to see what we see as robust biological signal from these. We have, though, run into issues where if we naively throw things into machine learning, it just memorizes the entire evolution of the genome or the species or of all the genes out there. And this is where we've started creating training and test data sets that are essentially evolutionarily guided or informed. And for one, we have a gene family guided training where we create training data sets based on certain sets of gene families, and then we test on unrelated gene families. The second approach is that we actually contrast orthologs with one another, and then we test on unrelated orthologs. And it's turned out that those two approaches of splitting testing and training for one another prevent us from overfitting and really just memorizing evolutionary patterns. And so Hai has used one of these models to ask the question, is it a pseudogene or is it highly expressed? He took the expression of maize genes across 66 different tissues, took the maximal expression across those. There's a group of genes we never see really expressed and a group that are highly expressed. And then he took about 1,500 base pairs of sequence from each end of the gene, ran it through the model, and created a CNN to look at that. And he essentially was able to especially between off versus highly expressed, was able to create a very accurate, high accuracy model between those two. And it did look like that most of the signal that was coming in from these models was coming from, it didn't really matter if we gave the promoter or the terminator, they were both providing some signal, and together both of them were improving the model. So we were able to predict whether gene was expressed pretty well just by training on those sequences. Then the one I think, at least from a quantitative genetics point of view, I've always been more interested in, if I have two alleles, I'd like to know whether or not the expression of allele A or allele B is higher. And that's the class of model that Jacob has been working on. And essentially, I'd want to know, does it go up, does it go down, or does it make no difference? And so that's what he's been training on. And the way he set it up was he compared orthologs. Some of these orthologs are between two separate species. And maize has a genome duplication, so we actually can use an ancient genome duplication. So we can even use the paralogs within maize as contrast in the model. So we've trained a bunch of different ways. Roughly, things work out the same way. We train on these, and essentially we take those, make convolutional neural nets from these, and we found out rather than trying to predict the difference, we tried to predict whether it just went up or down. And same style of model, except now you've got two orthologs in it, running through the convolutional neural net. And the interesting thing he was seeing there is that we only were able to do a really good job of prediction when the allele expression really shifted between the two paralogs, say by more than 10 percentile points in the expression. So the top 10% of differential expression, did we do a good job? But it turned out that we were getting most of the signal from the terminator sequence. And I'll show you where that was coming from. And so these are what are known as saliency maps, and they're highlighting what base pairs in the genome are really contributing the most, what base pairs in the model are contributing the most to the signal here. And what we see is in the ortholog one, where we're trying to predict which ortholog went up or down, it's the terminator sequence. Right before the terminator sequence is giving us the most signal in each of these models. While we're trying to predict the pseudogenes, most of the weight is coming in. We have weight coming in from the entire genome, but a lot of it is coming from the 5' UTR sequences are really helping us to determine what's being highly expressed and what isn't. And so when these two models are also kind of training on something different. The orthologs, these are still functional genes that have been conserved, and these are really much more a fine-tuning question. And it looks like the 3' UTR is perhaps really important for fine-tuning expression. It isn't determining what tissue is turned on or anything like that, but for determining how highly expressed that RNA is, we're seeing signal from that. We've also, Catherine's been also able to take her models and compare them against molecular assays where people have tweaked individual base pairs and done competition assays on them. And so she compared her regulatory probability model with competition assays and gets pretty good agreement between these. And so these are telling us that even single base pair changes, the models are sensitive to 1-2 base pair changes in the models and can start telling us what's likely to be going up or down. So what are some of the limitations of our current models? Well, I would say those deleterious mutation models that we were getting from conservation, our biggest problem there is that they were relatively good at splitting neutral from functional. So we could say that these 80 million SNPs don't matter, but these 4,000 do matter. We were not very good at saying within that last 4,000, which was the most important one. We were very poor at ranking within that last 4,000. Almost didn't appear to be had almost any signal from that. And a lot of that is because essentially we're comparing species with really divergent, and essentially selection has been able to do its job and purge all of those regions equally well from that. We were also not able to look at use conservation to look at regulatory landscapes very well. And the physiology of Arabidopsis versus maize is pretty dramatically different. So the rules of how the organism works are just simply not the same across that much evolutionary time. And so the route that we're focusing in on is, for example, 4 of these species were in the same tribe of grasses we call Dendropogonii. And we're starting to do saturation focus on a single clade. And so with my colleagues, Toby Kellogg, Matt Hufford, Jeff Rossi-Beyer, Adam Siepel, and Chi Sun, instead of just focusing on maize, where if we sequence 1,000 genomes, we get about 3 million years of evolutionary time out of that model. We've been able to learn a lot from that. If we sample 900 genomes from across the Dendropogonii, and they go back to about 18 million years of divergence, they sample together, the total evolutionary length across all those branches is about 1.5 billion years. And they, very importantly, they also share, there's a key evolutionary event back here that involves the development of NADP C4 photosynthesis. So this is one of the key evolutionary steps that allowed this group of grasses to radiate across the planet. They all share that, and then they also share roughly the same developmental plan. Maize looks different from everything else because of all that domestication, but everything else in the grass, if you weren't a trained botanist, you'd say, oh yeah, it's just a grass. And so they do share a fairly similar baseline physiology. And so one way to think about this is we're getting saturation evolutionary analysis. Essentially, every base pair that was in here that still persists today has been mutated at least 50 times, been mutated about 50 times. And so if it's still constrained or it's still conserved under certain environments, something must have kept it conserved. And we can directly measure that level of conservation because we're now tapping 1.5 billion years of evolution. And although they have a similar developmental plan and the same C4 photosynthesis, they are adapted to a tremendously wide range of different environments. And so we think that by tapping in saturation analysis on one clade like this, we'll be able to also mine out a lot more diverse fitness effects, adaptations. So where we are right now is I think evolution and machine learning really are providing some nice, working together very nicely. Evolution has given us near infinite mutagenesis. It's C-selection, we can see these fitness effects very well, and it's given us this big range of environmental history. But machine learning is allowing us to deal with these heterogeneous inputs at genome scale, and the learning can be really generalized fairly quickly to from one individual out. And so I think we're getting the tools at hand to identify these functional SNPs, and now what we're really learning how to do is really rank and to nominate given genetic variants. So what does our nomination process kind of look like right now? Right now we're nominating things like this, and then we're testing. We have lots of data on tens of thousands, hundreds of thousands of maize genotypes, and we can ask the question, do those groups of SNPs explain more genetic variants than we would expect by random chance, or a paired set of other variants? And if that model does work, then we can go ahead and essentially start nominating, editing away. But it is looking like the ability to edit genomic sequences is getting so efficient and so fast that we're going to be able to start nominating hundreds or thousands of SNPs, edit immediately, and essentially put them into uniform lines, and then directly phenotypically evaluate with either phenotypic evaluation or a GS-type model. So I think this is kind of the pipeline we're going to start to see in species like maize in the next couple of years. And I want to end there. And I want to highlight that I mentioned a couple of the postdocs who did some of the great research, but organizing a team like this and all those empirical data, Cynthia Romay, Peter Bradbury, and Sarah Miller have been really key in pulling all that together. And I'd be happy to take some questions.
Thank you, Ed. We're well ahead of time here.
Huh?
Yeah. So hope we've got plenty of questions. From Nick.
Hi. Sorry. So you talked about two quite different approaches there with the k-mers, which seemed like a fairly standard statistical approach, and then this deep learning convolutional neural nets, and you really, I felt a little bit like you only talked about why it isn't a black box for the first one. Do you really have any idea what's going on with the convolutional neural nets in these models? Because it seems like in a situation where you can just grow a whole bunch of maize and see what happens, and if it doesn't work, then no harm, no foul, then that's fine. But it does worry me quite a lot in other situations.
Yeah. No. So I think the question about the convolutional neural nets. So first off, a lot of the convolutional neural nets we're doing are not particularly deep, okay? So they're not like some of the image processing convolutional neural nets, which are exceedingly deep. And I would say what we're seeing with the saliency maps, they're giving us a pretty good idea of what base pairs and what regions really matter. And then and then it is very straightforward to start taking various motifs that we know about in these maps and start mutating them very systematically in the model to ask what happens. So we're taking things like hairpins and disrupting hairpins and seeing if that is affecting, is that what the model's been learning on? Has it been learning on other types of motifs that are out there? So I think we need to learn a lot more about how we probe the machine learning models. I totally agree for human medicine type purposes, the risk is way greater if you're wrong. Maize right now, I think we just need to have some belief to nominate towards the tail.
Hi. Sorry. Okay, so this whole approach of really finding out the mechanistic predictions for lots of individual genes and tweaking their expression and putting it together and making the super maze, that relies on really understanding how these effects combine and having a single selection target. Because in the end, you're going to have to test these strains in some sort of agricultural field condition. And then you wonder, is it worth the trouble? I mean, what's the cost benefit relative to simply keep selecting under, for total yield? Which is basically what you do.
Part of the problem is right now in maize breeding is introgression of a diverse allele brings along a lot of other regions, and these elite lines after, essentially you could kind of view it as $200 billion worth of R&D investment over a period of time are so good that we cross a land race in, it's a disaster.
Right.
Okay?
Yeah.
And so we want to take that knowledge that's in that land race and directly move it into this incredibly adapted, incredibly finely tuned genotype. And then we also need to do that for essentially, each of these has to be tuned for 1,000 different environments. So when we talk about elite maize, there's elite maize, but it's elite maize finely tuned for 1,000 separate environments. So and so once we make the edits, the field trials are not that expensive. So we can just select. That's not a particularly expensive thing to do. Deregulating them, depending on where you are, may be extremely expensive, but the actual field trial, if we can take a very elite line, tweak 1,000 things, and cross it back to the same line, and we segregate it, you could just apply phenotypic selection and move very quickly.
Can you just say a little bit more about the actual primary data you use? It's data from many different varieties. What are the actual measurements you're getting? Where do they come from?
Okay.
Because you need large numbers to do these deep learning things.
Right.
And it's not clear to me what the actual primary data are.
Yeah, so for example, on the expression data, so a lot of those are coming from just expressions, expression sequencing studies, where we've collected mRNA data from 60 or 70 different tissues. We've estimated expression at 37,000 different genes. We sometimes have these from 2 and 3 different reference genomes. And we have a quantified value for every one of those.
But the number of genomes you're looking at is limited then? Isn't it the number of genomes that should matter more than all the other stuff?
So,
or as much?
Sometimes.
Because you're really then sampling only a minority of the variation.
Yeah, but it does sometimes help to have enough evolutionary distance between alleles. Okay, so for example, we've been able to now train models between maize and sorghum. And so that gives us a fairly divergent range.
So how many samples of maize and sorghum do you use to look at that difference?
So some of those models just have the reference genome of just sorghum and the reference genome of just maize.
Isn't that rather risky? You're looking at so little of the overall variation that's there.
But the model is not, it's learning how expression is controlled across all the genes.
And you think that the genes don't affect the expression learning? The expression levels?
No, that's what we're predicting.
Question from Josephine.
So I take it in your funding proposals, you put up some ideas about how much gain you might make in all this. So if you edited 1,000 loci that were your top 1,000 in your machine learning, what would be the total effect size?
So in fact, we actually do not do that because essentially, being where we are in the U.S. system, there's a group of companies who are downstream of us who are really doing that. Okay. So we know what our yield is for our inbreds and what hybrids are able to produce. And so there's still a twofold difference between inbred yield and hybrid yield, or sometimes threefold. And so a substantial portion of that is probably deleterious mutations that are out there. And yield has continued to increase as inbred lines got better and better. So, any one of these genomes, we estimate, 1,000 or 2,000 deleterious amino acids that we can see pretty easily. I would say we're only right about that they're actually deleterious maybe half the time, because some of them are probably, the genes are becoming pseudogenes or other types of things, so they kind of, I wouldn't say we have that exactly right, but, and then I would say there's an equal number of regulatory ones. So how many of them we need to change? I mean, right now, we can't rank them very well. And so that's, we can say how much variance is being explained by them, and pretty much all of our models, deleterious variance explain about half the variance.
Okay, thank you.
So,
so you sort of implied that these deleterious mutations were recessive because you said, they're in the places where you see heterosis. Right. If they were completely recessive, then there's not much point in getting rid of them. Your heterosis works. But are you suggesting that they're only partially recessive? They're only partially recessive. And I think that's kind of the argument is, where are we along the partial one? I was just working with, visiting John Hickey's group, and that was kind of the question is, how recessive are they? But if you just look, over the last century of maize breeding, inbred line, heterosis actually has, percentage-wise, has dropped over the last century, and inbred lines just kept on getting better and better, and yield just keeps on getting better and better. So it's not completely recessive. So the heterosis has decreased over time. It's decreased over time.
from a percentage point of view. The absolute one is, but percentage-wise, is decreased substantially.
Could I comment on that?
Yeah, please.
I mean, there's an old literature, mainly coming out of Jim Crow's lab in Drosophila, which has shown that slightly deleterious mutations in Drosophila have dominance coefficients, of the order of 0.2, so far from being completely recessive. Right. And that's presumably most of the ones you're dealing with are relatively minor effect alleles. So,
yeah.
So complete recessivity, even lethal mutations, is a myth. Again, Jim Crow's lab showed that recessive lethals in Drosophila have an average fitness effect of about 2%. Mm-hmm. One of the problems is, plant people don't pay any attention to Drosophila. And neither do humans. If you want to learn about genetics, you should go to Drosophila.
Any more questions?
So a few years back, we sequenced and reassembled some Drosophila genomes, and we had gene expression data. So we found, because Arabidopsis, sorry, not Drosophila. I've got thrown off.
Sorry, Arabidopsis, yes?
It's a plant. Okay. So the point was that if you took the reference genome annotation as the gold standard, you vastly overpredicted deleterious mutations, because you essentially had a bunch of things happening which, for example, splice sites would change so that an apparent stop codon was skipped, something like that. Right. So I was wondering whether your machine learning is doing something similar, or whether you simply don't see this phenomenon at all in maize.
So that essentially it's learning...
It's learning the context so that you can say, actually, this stop codon isn't a stop codon, it's getting skipped.
I have no doubt that it's doing some of that. We actually were very surprised at the 3' UTR type signal that we got out of it. But now we're learning more about, for example, adenosine methylation in RNA and its regulatory importance. And so I think some of the things we're starting to see, once we saw that signal, and we started showing it around to molecular biologists, and actually I've talked to now a number of companies who've done tons of mutagenesis and, as they've engineered various transgenes, everybody kind of agrees, yeah, there's a lot of signal on regulation coming from 3' UTRs, and so we're now trying to tweak these models to see if we can identify, for example, the adenosine methylation sites that may be controlling the mRNA stabilities that are going on in some of them. And so then there's going to be hairpins and all sorts of other types of models.
I think we've got to leave it there, actually. We'll move on to our next speaker. Thank you, Fred. Mike, I'm sure you can ask that question later. So here we have Richard Martz. Richard's going to talk about Arabidopsis, not Drosophila. I can't pronounce it.